My paper talks about the effects of sex hormones and the EFMB3 gene in regulating blood pressure via the vascular smooth muscle cell contraction. So vascular smooth muscle cells can be found in a layer surrounding blood vessels. This layer is called the tunica media. And when these muscles contract, they reduce the diameter of the lumen, thereby restricting blood flow, thereby increasing blood pressure because the same volume of blood has to now travel through a much smaller surface area. And so therefore a stronger contraction ultimately translates. The investigators were also interested in looking into a receptor ligand combination. The receptor is known as erythropoietin producing human hepatocellular carcinoma receptor kinases, also known as EPH. They are considered the largest class of receptor tyrosine kinases, which can be further divided into A and B families. The ligands for these receptors are called ephrins, abbreviated EFN, and they can also be found on the cell surface and can also be divided into A and B subfamilies. And the difference between A and B is that they're bind to the cell surface differently. So in the diagram, we can see that ephrin A bind to the cell surface by GPI, and ephrin B's bind to the cell surface through a cytoplasmic domain. And the binding between the EPHs and EFNs are nonspecific, but typically ligands of subfamily B prefers to bind to receptors also from subfamily B. And these receptor ligand combos can be found in various different tissues of the body, such as the central nervous system, immune system, cardiovascular system, digestive system, and others. This paper specifically looks at the role of EPH and EFN in vascular smooth muscle cells. In particular, they are looking at the EFMB3 ligand in conjunction with the EPHB6 receptor, both from the subfamily B, and these both can be found in the smooth muscle cells. Previously, work has shown that some members of the EPHB and EFNB family can actually regulate blood pressure, specifically via the smooth muscle cell contractility. So in general, they knew the effect of the EPHB and EFNB proteins, but up until now, there hasn't been any studies linking sex hormones to the regulation of those two proteins. Therefore, one of the main purposes of this paper is to investigate the relationship of sex hormones to this blood pressure regulation in vascular smooth muscle cells via the EPH and EFN protein. So what happens when the EFNB3 gene is deleted? Well, at the start of the investigations, the researchers took a cohort of mice and knocked out the EFNB3 gene to observe its effects on blood pressure and heart rate, and compared that to the wild type group. For blood pressure, the mean arterial pressure, MAP, diastolic pressure, DP, and systolic pressure were taken. And generally, when blood pressure is described, it's given as the diastolic pressure over systolic pressure. And the mean arterial pressure is the average level of blood pressure. Before I get to the results, I just want to note that the x-axis shows the time course over 72 hours of observation, and the y-axis shows either pressure in millimeters of mercury for the blood pressure measurements or beats per minute for the heart rate measurements. The data points were analyzed using one-way ANOVA, and p-values are shown. The solid blue line represents the wild-type mice, and the solid orange line represents the knockout mice. The corresponding dotted lines are the standard error of the mean values, which tell us how accurate those measurements are to representing the population. These particular 4 panels represent the male cohort. And as you can see, with the overlapping blue and orange trend lines and a large p-value larger than 0.05, there is actually no change in all 3 blood pressure measurements between the wild type and the knockout mice. However, the heart rate in the knockout mice is statistically significantly higher to that of the wild type mice, supported with a p-value smaller than 0.05. Next up are the results after observing the female wild type and knockout mice. And these, as you can see, these results are completely opposite to that of the male mice. And as you can see, with the gaps between the trend lines and the small p-values less than 0.05, there's actually is a statistically significant increase in all 3 blood pressure measurements. However, heart rate was not different between the wild type and the knockout female mice. Lastly, they looked at vascular smooth muscle cell contractility by measuring the relative degree of contraction as a percentage, and they did this by stimulating a culture of vascular smooth muscle cells with phenylephrine, a vasoconstrictor, meaning that the drug causes smooth muscle cells to contract and thereby decreasing the diameter of the lumen. The figure is not shown, but the results showed that in male mice, contractility did not change between wild type and knockout mice, whereas in female mice, the knockout mice had an increased vascular. The next part of the experiment was to look at the effects of sex hormones on the EPH and EFN's effect on blood pressure and contractility, because if you think back to the last slide, male mice lacking the EFNB3 gene had no change in blood pressure or smooth muscle cell contractility, but females lacking the same gene had higher blood pressure and increased contractility. And so this set of experiments were performed in the same manner where the vascular smooth muscle cells were isolated from the female and male, knockout and wild type mice, and incubated with a vasoconstrictor for 15 minutes. And then a relative contraction was measured, as you can see with the y-axis. But the difference with this experiment is that these smooth muscle cells were treated with a sex hormone prior to adding the vasoconstrictor. And in order to prevent the carryover of sex hormones when isolating these living cells from in vivo to in vitro, the investigators treated the male smooth muscle cells with estrogen and the female smooth muscle cells with testosterone. In the first part of this experiment, as seen on panel A and C, the male and female wild type and knockout mice were incubated with its respective sex hormone for 3 days before the vasoconstrictor was added. In total, there were 4 treatment groups, 2 incubated with sex hormones and the other 2 control groups for both wild type and knockout mice. And in general, the gray circle represents the knockout mice treated with a sex hormone. The black circle line represents the wild type mice also treated with the same sex hormone. And the gray square line is the control group for the knockout mice, and the black square line is the other control group for the wild type mice. In panel A, we can see that estrogen increased the contractility of the knockout mice, and in panel C, testosterone actually showed a decrease in contractility back down to the same level as the wild type. And in general, the investigators are assuming that this effect would be the same in the smooth muscle cells in the opposite sex. Therefore, they concluded that in the absence of the EFN3B gene, estrogen could somehow be related to the increase of smooth muscle cell contractility, where testosterone somehow plays a role in decreasing contractility. And these panels suggest that in the absence of the gene, again, the sex hormones work to produce a response relating to smooth muscle cell contractility and therefore blood pressure regulation. Then the investigators want to test how these sex hormones regulate contractility, and one classification could be identifying whether or not they have a genomic or non-genomic effect. In general, if the response is genomic, that means that there's some signaling that triggers the regulation of gene expression of genes possibly relating to smooth muscle cell contraction. On the other hand, if the response is non-genomic, then there is some really quick process signaling that triggers contraction without having to turn genes on or off. And to test this research question, the investigators changed the incubation duration of the cells with the sex hormone, cutting it down from 3 days to a mere 15 minutes. And then they immediately added the vasoconstrictor. In panel B, we can see that estrogen still increased the smooth muscle cell contractility, which indicates that estrogen has some form of non-genomic effect. Contrastingly, in panel D, the short-term treatment of testosterone did not show the same decreasing contractility observed in panel C, thereby suggesting that testosterone has a genomic effect, by which it requires a longer time to actually produce a response. Overall, the data suggests that the EFMB3 gene typically has an inhibitory effect on the smooth muscle cell contractility, but acts in a sex-dependent manner. Lastly, I just want to quickly address an inconsistency with this data is that the testosterone is seen to decrease contractility here, but not in the previous experiment, where there was no change in contractility. One possible explanation for this is that the experiment here was performed only in female knockout mice smooth muscle cells, whereas the previous experiment looked at the cells of the male knockout mice. And what could be happening in males is that the EFN3B deletion will initially cause an increase in smooth muscle cell contractility, thereby increasing blood pressure. But testosterone, through some other mechanism, could decrease contractility, and so it opposes and counteracts the effect of the EFNB3 deletion, thereby producing a phenotype of no change in blood pressure or contractility. So that was a lot of information, and so I just made a slide consolidating all the important aspects and conclusions that I. So these are my questions, and thank you for listening.